we innovative neuroscience and impactful networking. And today we have the pleasure of having Dr. Carlos Aizenman as well as Dr. Linda Liscombe from Brown University. And today it will be a wonderful opportunity for all of us to not only to learn about the great science that is conducted at Brown, but also the academic and training opportunities they offer for our students. So let me introduce our speaker for this afternoon, Dr. Carlos Aizenman. Dr. Carlos Aizenman is professor of neuroscience at Brown University, and his interest in neuroscience actually began as an undergraduate at Brown, where he worked in visual cortical synaptic plasticity in the laboratory of Dr. Mark Beer. As a PhD student at Hopkins with Dr. David Linden, he studied plasticity of inhibitory inputs and of intrinsic excitability of deep cerebellar nuclear neurons. His postdoctoral work was done at the lab of Hollis Klein at Cold Spring Harbor, where he continued his interest in the visual system, in particular in the regulation of neuronal excitability. Now, at Brown, his studies focus on how early experience shapes developmental sensory circuits and how this regulation of these processes leads to neurodevelopmental disorders. And because of his influential and impactful research, Dr. Aizenman was selected as one of the 100 most inspiring Hispanic Latin scientists working in the United States. So for me, it's a pleasure to introduce Dr. Aizenman. Carlos, thank you for being here. And yeah, let's hear about great science. Thank you.
Thank you, and thank you for the invite and everyone for coming. Let's see, let me get this going. And it's okay. So let me just unplug my other monitor because I think this only. Okay. So everyone can see?
Yes,
okay.
We are.
Good. So I only see my full screen. So, all right. So I'm going to tell you a little bit today about a project that we've been working on in my lab, maybe over the last 5 years or so. It started about 5 years ago. And I'll tell you about some of the latest findings from that project, which is the role of MMP9 in neurodevelopmental disorders. So before I start, I just wanted to acknowledge all the people in my lab that have worked in this project. It was started by Arseni and Eric, who decided to study autism in tadpoles. I'll tell you a little bit more about that. And then it was all these people have contributed to some degree. A lot of the focus on MMP9 was done by Siley, who's a current postdoc in my lab. But you'll see a little bit of everybody's work in here. Okay, so if you talk to a doctor or a clinician, sorry, I'm putting on a timer because again I can only see one screen. Okay, if you talk to a doctor or a clinician, and you ask them what autism is, they'll tell you, oh well, it's a disorder of development that's characterized by two main symptoms. One of them is disorders in social communication and interaction, and another is restricted and repetitive behaviors. And then the interaction of the two is usually when you get a diagnosis of autism. I'm sorry, the phone is ringing and nobody in my house is answering. Hopefully they will soon. Okay. But this is not a particularly useful way of understanding autism. It's because it doesn't really tell you anything about the disorder or the cause of it. And one way that's more useful to think about it is from this lens of neurodiversity. And so if you take this curve and you look at the normal variation in human brain function in any sort of modality, like cognitive ability or motor processes or vision or any sort of thing, language, anything that's produced out of the human brain, people are all going to fall within a certain range. Most people are going to fall within a middle range, but then there's always going to be some people who are on the outliers of this. And if you take several of these variations in human brain function, you could create some kind of multi-dimensional matrix if you want. But there'll be some individuals that will be on these edges of the curve. And one useful way to think of it, well, what does it autism diagnosis mean? Well, it just means that you're on these extremes past a certain threshold. But what's important is that we're not talking about a different process or a fundamentally different brain. Rather, it's just the edge of a normal continuum, right? So by understanding the variability in normal brain function among individuals and what makes us all fall within this range, we can get a better understanding of how several neurodevelopmental disorders come about and understand a little bit about their mechanism. So one useful way to think about it too is to think, well, what are some of the other neurological manifestations that are associated with ASD? And so in about 20 to 30%, there's epileptic seizures of people with autism. At some point in their life, they might have a seizure. There's sensory integration problems and hypersensitivity. This is fairly common. Often there's problems with motor coordination and to some degree of intellectual disability. And actually, this should be revised. It's much lower than 40%. And this could also be extremely mild or extremely impaired. And then it's also useful to think about co-occurring conditions like Fragile X, tuberous sclerosis, Angelman syndrome, that co-occur with autism as well. And so if you try to come up with what's happening at a lower level in terms of how the brain is organized, it's useful to start looking at the genes that have been associated with autism spectrum disorder. And it turns out that there was a paper a year or two ago that identified at least 100 different genes that are associated with autism. These are some that have been studied in our model organism in Xenopus, but you can see that there's a lot. And so the interesting question is, well, how do you go from having 100 different possible genetic loci or mutations to something that we all recognize as one disorder or a spectrum of disorders? How do you get from such a diversity of genetic causes and to some degree interactions between genes and environment to a relatively limited set of symptoms? And so if we start studying these lists, it turns out that a lot of them are actually genes related to synaptic transmission and to signaling at the synapse, as well as regulation of gene expression, right? So that suggests that autism spectrum disorder is something that arises from some low-level dysfunction at the level of synaptic transmission. And what's interesting is that when we look at these variations in autism-associated genes across the whole range of studies, in many cases it's an upregulation of the gene or like a gain of function. Sometimes it's a decrease in the gene. And so a lot of these genetic elements occur within a specific range, and then any deviations outside of it will then start creating some small effect that can cascade into something that we recognize as autism. Okay, so one of the fundamental questions that we're interested in is how does synaptic dysfunction during development lead to neurodevelopmental disorders? So, well, if we're talking about autism, that's a uniquely human manifestation of low-level neurological deficits, right? Only humans can be autistic. You can't have autistic animals, and that's because we define autism based on a series of very human characteristics such as language and social communication in ways that are unique to humans. But really, to get a good understanding, we need to look at multiple levels of analysis. really from synapses, which is where we think a lot of the dysfunction lies, all the way to behavior. And so the only way to do this is in an experiment, is to look for an experimentally tractable and accessible model organism. And then the more animal or models that we use, we can then start for looking for intersections between the phenotypes across multiple experimental models. And so if we find that there's something that occurs in common, if you say do a perturbation in a certain gene and you see that there's a certain effect that's in common in a mouse or in a zebrafish or in a xenopus or in a fly, that's likely to be an important locus of intersections where it might be something that might tell you a little bit more mechanistically about the cause of the disorder you're interested in. So keeping this in mind, in my lab, we've chosen to use the Xenopus laevis tadpole to model neurodevelopmental disorders in a way that would be analogous to looking at autism, but in a model organism. And the nice thing about this experimental preparation is that we can focus all the way from the level of synapses to individual neurons, to looking at wiring by looking at activities of neural networks, to general connectivity in neuroanatomical pathways, and then we can also look at behavior. So if we induce a perturbation here at the synaptic level, we can see how it can cascade all the way up into behavior and how these different steps change over time. So the way that we've started to model, that we started initially to model neurodevelopmental disorders in Xenopus is by giving them exposure to valproic acid. So VPA is a commonly prescribed anti-epileptic drug. It has some mood stabilizing effects as well. And while generally considered safe as a drug, it's been shown to have some effects on some deleterious neurodevelopmental effects, especially when there's prenatal exposure. So in women who were being treated for seizures while they were pregnant, about 12 to 15% of their children and they were taking VPA, they're diagnosed with a neurodevelopmental disorder by age 6, which is much higher than the general prevalence, even in women who suffer from seizures who are not treated. That's only around 2%. And if you look at these 12 to 15%, about 80% of these are autism spectrum disorder. So it's something that increases the likelihood of developing autism quite a bit if you have in utero exposure. And so this drug is no longer taken when someone is pregnant, but there were some retrospective studies. The biggest one that came out around 2013, and I think there was an even slightly newer one. Okay. So, oops. Okay. So to do this in tadpoles, we decided we exposed tadpoles to VPA in the rearing media during a critical developmental period. So that part of the tadpole brain that we use as a readout for what's going on in the rest of the brain is called the Optic Tectum. And the tadpoles goes through several developmental stages. And we start to study this circuitry, which is the Optic Tectum is a midbrain structure that in mammals it's equivalent to the superior colliculus. And it's one of the primary sensory areas in the frog brain. It receives multisensory inputs from many modalities, but one of the biggest projections is from the retina. And we start to study these animals from the time that the retinal axons have already innervated the tectum, and then there's a period of growth where the recipient cells in the tectum and the axons from the eye proliferate, and then there's a refinement where there's axon and dendrite reorganization and pruning of inappropriate axons, such that you get a much more refined retinotectal map. And this is an activity-dependent process. Is thought to be mediated by Hebbian plasticity as well as various permissive cues that are driven by activity. So what we did is we exposed tadpoles to VPA. They soak everything that's in their media because they don't have a very good blood-brain barrier. And so they're exposed to the drug for a period of about 8 to 10 days when there's this big amount of refinement and sculpting in their brain. Then we either do behavior or electrophysiology to see how these different parameters are affected. And so the first thing we looked at was a behavior that's a schooling behavior. And you could see tadpoles here forming a school, forming various schools. They're swimming in a bowl, and they tend to congregate into small groups, and they tend to swim in the same direction and form these sort of schools. They're not quite as automatic as you would see in fish, where the schools are very tight, but rather they tend to form and unform over time. And so we can quantify this by looking at the distribution of tadpoles on a dish and looking at the distances between the animals and comparing ones that have been reared under control conditions and ones that are reared in VPA. Now, this is just a cartoon, but it kind of tells you a little bit about the data we get. So this is an actual diagram of the location of the tadpoles and the inter-tadpole distances. And notice that on controls, you find these short distances between tadpoles in schools and then some long distances between the schools. You can plot these and you see there's a little bit of a biphasic distribution where you see the schools and then the short distances and the longer distances. In the VPA-treated tadpoles, there's a shift to more sort of intermediate distances, right? So there's a disruption of this schooling pattern. It's easier to see if you look at that direction. They always tend to swim in the same direction as their neighbors. So you can look at the inter-tadpole distances to their nearest neighbors and plot those. And normally they all tend to be swimming in the same direction as their neighbors, some of them opposite direction. In the VPA-treated ones, there's more of an even distribution where tadpoles aren't quite. They're more or less swimming in random directions. So this told us that this schooling behavior was disrupted. And this is an interesting behavior because it has both a more or less social component where they have to find their co-specifics, find other tadpoles, and this is thought to be mediated by olfactory cues. And then there's a multi-sensory integration component where they need to kind of orient themselves in the same directions as their neighbors, right? They need to kind of see where the other tadpoles are. They sense a vibration through the lateral line, and they orient themselves to form this school. So it has multiple sensory and social components in it. And it's a very sensitive assay and very reproducible. Even though these effects aren't dramatic, I think that that's sort of what we were trying to do, is that you don't want to create a perturbation where the animal is just sitting there or there's something wrong with the brain. You want to create something, you want to create an effect that is subtle, right? Because again, something like autism spectrum disorder is something that is normally very subtle, right? 90-something% of brain function is fine, right? It's just that there's these things that we're very attuned to, like language and communication, that we notice differences. But in terms of the basic brain function, you can't really tell much difference in this case. And so in a way, it's a nice model because it's a subtle effect, it's very sensitive. Okay. So then we did electrophysiology to see how what's different. And you can do what's called an open brain preparation, where you can remove the entire brain. The tadpoles are very small. The brain is maybe 3 millimeters long, and you can fillet it open, lay it in a dish, keep it alive, and then stimulate the optic nerve and do whole cell recordings from cells in the optic tectum. And you can record visual responses as well as intrinsic firing in these cells. So this is kind of like a summary of many findings, but one thing that jumped out at us is that in general, in tadpoles that had been treated with VPA, there's a lot more synaptic transmission. So these are spontaneous excitatory postsynaptic potentials and inhibitory potentials. which we've separated by holding the membrane potential at different levels, so we can see excitation and inhibition. And if you look at the frequency of these spontaneously occurring events, so remember, you have a whole brain, and periodically synapses will fire, and you see these spontaneous events. There's more in the VPA-treated tadpoles. This is quantified here. Again, the frequency of the spontaneous EPSCs and iPSCs is higher in the VPA. And again, it's not a huge effect, but it's something that's very reproducible and ends up being very significant. So that means that there's you could think about it as there being more synapses. The amplitude of these spontaneous events doesn't seem to change. So that means that the synapses themselves look normal. There just seems to be more of them in the VPA-treated tadpoles. So we wanted to figure out, well, what's the source of some of those synapses, right? Because if these are the cells in the optic tectum and this is the input from the optic nerve, there's also, aside from these visual inputs, there's a lot of local connectivity. And this local connectivity corresponds to a lot of the circuitry that's within the tectum. They connect to each other. And this is obviously very simplified, but it just gives you a sense that there's local axons that can be activated in a feed-forward manner or feedback manner. Both when you stimulate the optic nerve, these cells fire and will then activate each other. And so you can look at this local network activity by giving a strong stimulus and looking at this polysynaptic activity. So these are young tadpoles, stage 44, 46. And these are just all controls. If you stimulate, this is where you stimulate the optic nerve. This first little peak is a visual input, so it's not very big. And then there's all this polysynaptic network activity. You trigger all of this local activity. As the tadpoles mature through this about 8 to 10 day period, the visual inputs get stronger, and all this local connectivity gets pruned away and becomes much more temporally compact. And this is a process that is guided by the patterns of activity, and there's a whole other interesting story about that. What we found is when we treated tadpoles with VPA, when we recorded at these later stages, stage 49, we found that all this local activity is still there. They look like immature tadpoles, right? This is a control mature. It's almost no recurrent activity. With VPA, there's all this polysynaptic activity that does not get pruned away. You can integrate the charge under the curve and measure it, and it's a significantly bigger effect. Also in the VPA, you get these kind of spontaneous bursts of activity. If you just record, you'll see that there's all these kind of network events. This is just spontaneous recordings for a period of about 20 seconds, and you can see that there's these what we call barrages, and these are just the number of barrages per minute, and they occur occasionally in some controls, but they tend to occur a lot more frequently in the VPA. So that gives us a sense that we see a lot more sort of local and recurrent connectivity in our VPA-treated tadpoles. So what we decided to then see is to see, well, can we find a behavior that might be sensitive to this local connectivity? And looking at these responses reminded us a little bit of seizures, right? It almost looks like there's these network events that could be sort of underlying some kind of seizure. And so we decided to test their seizure susceptibility by exposing them to a convulsant, which is PTZ. It's a GABA blocker. And if you just put PTZ in the control media, you can induce seizures. So this is just the tadpole swimming on a dish. So this is without PTZ. And then if you give them PTZ, about 10 minutes later, then they start having seizures. They have these periods where they're lying down on the ground paralyzed, and they swim quickly. Then they form those C-bends that you see where the head and the tail touch each other. And so right now the tadpole's having a seizure. And then he's going to stop. And then he's going to go into another seizure period. There. So what we can do is we can track the movement of the tadpole while they're being exposed to PTZ. And we can compare the control tadpoles with the VPA treated ones. And so this trace just represents the movement of a tadpole. Here's when we add the drug, and you can see that gradually you get these increases in speed, which represent the seizures, and then eventually these seizures become really frequent. And so we can just count how many of these seizures we have, and to get the seizure frequency, it obviously gets higher as time goes by. But this is over a period of 20 minutes. And in the VPA-treated ones, there's an increase in seizure frequency, suggesting that all this local connectivity that we found makes the tadpoles more susceptible to seizures. Another behavior that we looked at is startle habituation. So we can have a protocol where we put tadpoles in a dish and we play a series of auditory clicks and we measure their startle response. And we do this over, every 2 seconds or so, or 5 seconds or so. And you'll see that if you measure the startle speed, if you look at these blue lines, they'll adapt. So there's rapid habituation. But then when you start the next bout, 5 minutes later, they're already adapted. They remain habituated. And this kind of short-term, what we call short-term habituation, remains over time, right? They sort of remember between bouts to habituate. Even if you wait 15 minutes and come back, they're still habituated when they start. And that's what we see. This is a long-term habituation and the short-term. The VPA, there's still a little bit of very rapid habituation during a bout, but between bouts, there's almost like a bit of sensitization. They always start in the non-habituated state. So there's no short-term habituation and no long-term habituation. That means that the tadpoles, remain startleable, right? They have the sensory hypersensitivity that the controls habituate to, which is again something consistent with having too much recurrent circuitry that might mediate the startle response. We also know that both seizures and habituation involve the circuits in the tectum, although they're probably also involving other parts of the brain. And it's not surprising because VPA is affecting the whole brain, not just the tectum. We're just using the tectum as a readout. Okay. So that was there's a little bit more to that story, but that gives you the basics. That tells us that we have a nice way to model neurodevelopmental disorders similar to autism in tadpoles by exposing them to VPA. So we wanted to see next, well, okay, so now we have a model and something that causes something. Now, how does that work? What is VPA doing, and what are some of the interesting downstream things? So VPA, it works to inhibit seizures when given acutely because it inhibits GABA transaminase, which is the enzyme that breaks down GABA. So you have more GABA, and it serves as to prevent seizures acutely. Now, over long-term exposure, which is what we do, valproate is also a histone deacetylase inhibitor. And what that does is normally DNA is wrapped up in chromatin in the nucleus, and chromatin is deacetylated when in histones. When the histones get acetylated, the histones unwrap, and then you get increases in gene expression. And this process of acetylation and deacetylation is something that is tightly controlled to control gene expression. When you inhibit these HDACs, when you inhibit the histone deacetylases, the chromatin basically gets stuck in the state, and so you get overall dysregulation, long-term dysregulation of gene expression over a key critical developmental period. So Chris Charleglio in my lab, he did some microarrays a few years ago looking at tadpole, looking at Tectum from tadpoles exposed to VPA, and looked at all the genes that change. And it turns out that in the VPA, once, about half the genes increase their expression, half of them decrease their expression. So there's obviously a big, profound change in gene expression. And by looking through this list for genes related to development or related to activity, one that really jumped out at us was this one called MMP9, which is a matrix metalloprotease 9. Now, we found, confirmed using qPCR that if you expose tadpoles to VPA, you get increases in MMP9. If you use a different histone deacetylase inhibitor called SAHA, there's even a bigger increase in MMP9. So what does MMP9 does, and why is it interesting? Well, so for one, it's a matrix metalloprotease. It's a zinc-dependent metalloprotease that's transcribed locally and it's translated locally, and its translation at dendritic spines or synapses, and its translation is dependent on neural activity. It gets secreted in a pro form that then gets cleaved into an active form, and then this matrix metalloprotease starts cleaving all sorts of different targets in the extracellular matrix. That's what they do. And one of the important roles that have been proposed for this is that it creates a permissive environment for developmental plasticity, right? It starts cleaving multiple targets that allow neural circuits to rearrange during development. So, for example, there's these nice studies from Betsy Quinlan where they can reintroduce the critical period in adult animals by putting them in the dark, and somehow that causes increase in MMP9 expression, creates this new permissive environment, and now they can restart critical period in adults. So it's something that promotes plasticity. Okay, so we thought, if VPA is working through MMP9, we can make two predictions. One is, does MMP9 overexpression copy the effects of VPA? And the other is, does blocking MMP9 activity rescue the effects of VPA? So first, we overexpressed it. We created a plasmid and used whole brain electroporation to introduce MMP9 into the tactile neurons. It's also co-expressed with a fluorescent protein. So we could record from overexpressing cells. And like with VPA expression, if you record from these MMP9 overexpressing neurons, you see an increase in spontaneous EPSC and iPSC frequency. So again, you increase the number of synapses compared to ones that are expressing GFP alone. There's a little bit of an effect on untransfected neighbors, which is not surprising because this is a secreted protein. But the amplitude of these spontaneous events was not affected. If we look at these spontaneous barrages of activity and count how many we see that the ones that were transfected with MMP9, we see there's an increase of them in those brains, but not in the controls. Again, nearby neighbors obviously see the barrages as well because it's a whole network event. So again, increase in synaptic transmission and local connectivity. So does blocking MMP9 reverse the effects of VPA? So here is some spontaneous EPSCs, and these are evoked responses where we see recurrent activity. If you expose them to VPA, there's an increase in synaptic transmission and local connectivity that I just showed you of recurrent activity. If you treat them with a drug called SB3CT, which is an inhibitor of MMP9, while they're being exposed to VPA, so you get an increase in MMP9, but the MMP9 is inhibited, then there is no increase in spontaneous transmission, right? We see that these are not different at all. And likewise, you prevent this lack of pruning of local connectivity. So again, we can rescue the effects of VPA by inhibiting MMP9. Now, the other way we inhibit MMP9 is to inhibit its translation. So we can use what's called a morpholino oligo, which is an antisense oligo that is stable. So you can transfect it into neurons. And that basically binds to the mRNA and prevents it from being translated. And so you get a damping down. It's not a complete elimination of the protein, but you get a damping down of the protein levels. And if you do that and then expose them to VPA, again, you reverse this increase in synaptic transmission and in evoked responses, but there's no effect on amplitude of these events. Now, what's interesting is that if you just inhibit MMP9 in control animals, not animals that have been treated with VPA, I keep looking at my watch, so I'm not late. If you keep treating them, if you treat control animals, there is no decrease in basal transmission. So So what this tells us is that somehow VPA causes an elevation of MMP9 levels, that if you inhibit those, then you can reverse the effects, but basal levels of MMP9 are likely to be low. And we'll get to that in a second. Now, what about behavior? So I showed you that there's normal habituation in animals to startle. VPA show no habituation. If you rear them with the MMP9 inhibitor, you can rescue at least partially the habituation. Likewise, there's an increase in seizure susceptibility with VPA that can be reversed when you co-expose to the MMP9 inhibitor. So we see that inhibiting MMP9 rescues both the synaptic and the behavioral effects of VPA, which we were pretty excited about. And so we started to think, well, what is this? What does the MMP9 then do normally, right? We know that VPA creates this unusual situation where MMP9 levels are elevated over development, and then they cause all sorts of trouble. But if inhibiting MMP9 under control conditions doesn't do anything, when is MMP9 ever used in development? And we thought, well, maybe MMP9 is recruited when there's a big plasticity event, when there's lots of activity that requires structural plasticity. It triggers the expression and activation of MMP9 and creates an environment that's permissive for the structural plasticity. So we decided to use an assay for activity-dependent structural plasticity and see if we could modulate it by modulating MMP9. So nice thing about Xenopus is that they're transparent. So you can do single-cell electroporation where you can look at a single neuron in vivo using fluorescent marker. And you can image the same cell over periods of time to see how the dendrites grow and change. And so in Holly Klein's lab, they developed a protocol, an assay to stimulate dendritic growth by putting animals in a chamber where there's flashing lights everywhere for about 3 to 4 hours. And so what you do is you transfect a single neuron, take an image, put him in the dark for 3 hours, take another image, and then put him in the flashing lights for 3 hours and take another image of the cell. And you can compare the growth in this dendritic arbor. And so this is the growth rate in the dark, in the first image to the second image. There's usually not much change. But then when you put him in the light box with the flashing lights, there's always an increase in dendritic length. If you compare these to ones that were exposed to the MMP9 inhibitor and put them in the light box, there is no increase in growth rate, right? You can look at the changing growth rate in their controls and in the MMP9 inhibited ones. And so blocking MMP9 blocks activity-dependent plasticity. The other question was like, well, does this exposure to light increase levels of MMP9? So this was from a series of Western blots that were done in Holly Klein's lab. And there is an increase in MMP9 levels when tadpoles are exposed to these several hours of flashing lights compared to controls, compared to just being either in the dark or being just in the counter in normal light. So the model we have is that, again, VPA is increasing the transcription of MMP9 and or translation. And then this leads to hyperconnectivity, leads to too much plasticity. So if you just overexpress MMP9 alone, you also get this hyperconnectivity. If you inhibit MMP9 while you're exposing to VPA, then you don't get the hyperconnectivity. Normally, when there's activity, MMP9 levels are elevated, and then that leads to structural plasticity. If you inhibit MMP9, then activity does not lead to structural plasticity. So that we thought was very interesting, especially because there's been a lot of focus on extracellular matrix lately and the role of these metalloproteases, these perineural nets that are thought to develop after the critical period are thought to stabilize a lot of the neural circuitry in later development. And these MMPs are thought to be able to chew this up, these perineural nets that surround several neurons, and then facilitate plasticity again. So this kind of adds to this knowledge and also allows us to think, well, how you could get this hyperconnectivity, which is something that is a phenotype in a lot of these models of autism, right? That you lead to too much connectivity, which can lead to sensory hypersensitivity, seizures, and other behavioral phenotypes. So what I'll tell you about the last thing that we're doing, which will take 1 minute. We started looking at the structure of the dendrites in after VPA exposure, and these are reconstructions using fluorescence, just cells expressing GFP in controls and VPA treated tadpoles. And while there is no big exuberant growth in dendritic arbors, which is what we were expecting, there's a large redistribution of dendritic branches. So there's a normal sort of sprouting of branches where you normally wouldn't see them, right? And this is a shoal analysis showing you the density of branches as a function of distance from the soma. Normally, they all kind of branch around the middle, but then in the VPA ones, they're sort of branching all along the arbor. And so Audrey Medeiros, she did a rotation in my lab and wanted to try out Brown's new serial block face EM microscope. And so she prepared a block of tissue from controls and went from a VPA tadpole of the Tectum. And this allows us to start reconstructing some of these neurons and their axons in extreme detail. We could see the location of every synapse and every axon that's connecting to the cell. And this is a control neuron. The light blue is a dendrite. And you can see that these axons tend to be layered in layers. And this is known, it's known that the different sensory modalities innervate at different layers. Some of these are probably local axons, but a lot of these en passant axons that pass through are sensory fibers. If you compare this to, and this is something that several undergrads, Alyssa Rust and Blanca Perez, are following up in the lab. And if you compare this to a VPA one, you see that the distribution and pattern of axons is all over the place. There's none of this layering that we see and that we normally see in the others, and the axons look skinnier for some reason. And so right now we're trying to get quantitative data of this. It takes a very long time because we're basically doing this by hand. I mean, we have software, but the software does not do this automatically, so it's very time consuming to reconstruct these. But if you notice, this is, for example, from a VPA one, there's all these little filopodia that are growing out of the dendrite, and all these synapses that in the controls are normally clustered together, they seem to occur all over the arbor, and then there's these kind of sproutings of growth suggesting some disturbance in the extracellular matrix. So this data will be interesting. It's great because it can be done remotely, so we've been working a lot on this over the last few months. Okay, so using this comparative approach can give you a fundamental understanding of the biological deficits that lead to differences in brain function. This integrative approach tells you how a perturbation can cascade to the system and bring you from low-level deficits to behavioral phenotypes. And many of these perturbations ultimately converge on similar behavioral effects. And that's why you can get such a diversity of genes that are associated with ASD. So these are all the people in the lab, past and present, external agencies that have funded us, as well as Brown University, that have funded this particular work. And that's it. Thank you.
Thank you, Carlos. Gracias. We have a couple questions for you. The first one from Karima Costa. She says, why did you choose to study sensory area instead of the areas involved with motor outputs?
Well, because I think usually, so this spinal cord, for example, there's not as much plasticity, right? And as much susceptibility to change. And so we wouldn't necessarily expect to see some changes there. Now, we could have looked at some sensory nuclei in the hindbrain, but, you know, we have 10 years of data of looking at this area, which is not purely a sensory area. It's sort of like a sensory motor way station that then gives motor output. And so it would be interesting to follow up studies in the hindbrain, but it's something that we would have to start from scratch, including figuring out what the circuitry is. Because of our lab is also interested in multisensory integration and a whole bunch of other things related to sensory processing, we figured that that was a good area to focus in. And, we think that there's probably things going on in motor areas per se, but this, as I said before, the Tectum is a readout for whatever else is going on in the brain. But that's a really good question.
Okay, good. We have another question from Cristina Velázquez. She says, so does anti-seizure medication cause seizures in Tadpole?
No, so that's a little, so that can be confusing. So the anti-seizure medication doesn't cause the seizures. What happens is that if you have a long-term exposure, the side effects of the anti-seizure medication cause an increase in connectivity that enhances the seizure susceptibility. So when we're testing them, the anti-seizure medication is already washed out, right? So it's not there, but it's almost like a side effect that we're interested in. And so, yeah.
Okay.
So, one thing we want to try is to just see if could it be also a response to suppressing GABA transmission or enhancing GABA transmission over a long period of time? Do you get a compensatory increase in circuitry? So that's something that we're interested in testing.
Great. And then there's another, are the animals put in those different lightning scenarios in development?
So that's an excellent question. So in this experiment, it was just acute, right? 4 hours, but we are doing, we've done some experiments where we change the sensory environment for several days or a week and look at the effects. And that has a profound effect on sensory processing. We haven't looked at it in the context of seizures, but definitely in the context of multisensory integration.
Okay. And there's another question says, can you comment on what are the defining factors that determine whether an anticonvulsant leads to seizures or inhibits them? Is it a function of developmental stage, you think, or?
I mean, I think acutely it will inhibit the seizures, but I think giving chronic exposure over development is what then starts to promote the seizure supporting circuitry. So I think what it is the length of exposure and the amount of plasticity in the brain.
And so what would be the underlying molecular features that lead to that behavioral phenotype?
Well, so in this case, I think the molecular feature is that we're altering gene expression over long periods of time and dysregulating several genes that are responsible for plasticity.
Okay. And finally, we have another question says, how did you come to choose tadpoles as a test subject?
Well, that's interesting. So after doing a PhD working on the deep cerebellar nucleus and realizing that it was incredibly difficult and frustrating and I never wanted to do it again, I went to Holly Klein's lab where they had this excellent model where you could, again, as I said, go from synapses to look at cells in vivo, and you could do all these things. And then I started doing electrophysiology there, and it was like 800 million times easier than in anything else. And you could get so much data and test so many things that I said, this is a perfect model because you have all the same brain regions, all the same proteins and genes. The brain is organized in the same way because it's a vertebrate. So why not? It's almost you can really just, if you have a crazy idea, you can just test it and you'll know within 2 weeks whether it's going to work or not. So it's a great preparation for modeling, for testing ideas, and for just getting a whole integrative view of development. So the plan initially when I started my lab was to switch to mice or something, but then 15 years later we're still getting a lot of good data, and so as long as we can get the data that we want and it seems to be the best preparation for the questions we're interested in answering, we'll continue to use Xenopus.
Sounds good to me. It's wonderful, beautiful data, especially what it struck interesting is how the disarray of the connections for the phenotype of the considered for the autism. It's so I should say cellularly consistent and relevant to what you see in behavior.
Yeah.
It's really amazing, and what you just presented, it just makes total sense to me. Someone that is very interested in the topic, but of course, as we all know, it's a spectrum disorder, and it's a range, and it's quite complex because you have language, social behaviors, sensory integration, and all sorts of things that if you do an MRI of the brain of a autistic individual, you want to see if there's a difference, and then you won't see anything.
Right.
Like so it's something so intrinsic of the connectivity that you just showed that is the amazing part of it. So
and that's what I always gonna say, that if you're an alien from outer space and you come to Earth and you see some people who are autistic and some who are not, and you look at them and you're sitting there, it's like, well, you won't notice a difference, right? Because they're walking around, they're eating, they're sitting down, they're talking, and so the difference is very, 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 very small. We're just so attuned to social cues that we can notice something, but that's just because our brains are sort of wired to look for these things. But from an external point of view, the differences are very, very, very small. As you say, you do an MRI, you were not going to find much.
Yep. Well, thank you, Carlos. It was a wonderful talk. I really appreciate it. So now we go to the other aspect of our seminar, which is the part where we can get to know a little bit about the training and graduate program at Brown. And for that, we have Dr. Lipscomb and Nadira Youssef, which is a graduate student and also a UPR alumni. And so Nadira will also be there for answering questions or commenting on her experience at Brown. But first, we'll have Dr. Lipscomb to talk. Diane?
Yeah, thank you, Carmen. Hi, everyone. And I'm really glad to have Nadira here. You can hear me fine, right? Yes.
Yes.
Okay. Actually, Nadira, I think that's the first time you never asked a question in a seminar.
Right, that's true. It's like, it's supposed to be like proper now, right?
All right. So, look, I think, Carlos and Nadira and I want to have this be informal, but I thought I'd just have a few slides to show you. But really, the point is for you to ask questions. And let's see so Carlos, I should go into presenter mode, right?
Where it says present now,
okay. Yes, somewhere. Oh yeah, present now, and then I should go to the full screen. Okay, great. So my entire screen, great.
Yep.
And then let's present this. Okay, great. And Nadira, I want you to yell at me if I go more than 10 minutes. Okay.
Perfect.
Great. Yeah. So I'm going to give you a quick run through about the neuroscience graduate program at Brown, but also to tell you a bit about what we care about, and just to mention, you know Nadira, and she's going to talk more, but we have Roberto, who's in a graduate program that I'll talk about in a second, and Gabby, our really wonderful colleagues from Puerto Rico.
Diane, I don't think we. Yep. Are you supposed to have a slide on right now?
Yes.
We can't see it.
Oh, okay. Let's go back. So
present now.
So present. Okay, hold on. So share. Okay, I'm sorry. Should have done share. Let's see. Cancel. So so you can see my screen though, right?
No.
Not yet.
Oh, you can't see anything. Oh, okay.
I can.
Right, let's cancel. Let's try one more time. If it doesn't work, it's fine. I can just.
I have, yeah.
Or do you want to just? Let me just see if I can do it. So should I do entire screen for present?
Yes.
Or a window? Okay, entire screen.
Either or.
But it doesn't. It just says share your entire screen. Chrome wants to share the contents.
Yeah.
Choose what you want to share. Oh, okay. Here we go. Share. Stupid me. All right, there you go.
Okay, good.
Okay.
Yes. All right, great. So here we go. So
yeah.
Now you can see our amazing friends from Puerto Rico, and I already introduced them. And so moving on. So where are we? And this is Rhode Island. And actually, this is the Native American map of Rhode Island. So we're the Narragansett tribe and the Wampanoag tribe are really the natives of this area. It's a beautiful area of the country. And I thought I would do just a little kind of comparison between Rhode Island and Puerto Rico so that I can learn more about. I've been to San Juan. It was absolutely, completely beautiful. I loved it. But the nickname of Rhode Island is the Ocean State because there's a lot of ocean around. And Borica, is that right? Correct pronunciation?
Yeah, so Puerto Rican people call ourselves Boricuas because the original name of the island was Borinquen.
Yes, great. Okay, so I heard that pina colada was invented in Puerto Rico, and Rhode Island invented coffee milk. I don't know what that comparison means, but it's kind of interesting. Puerto Rico is about 3 times the size of Rhode Island, and we're the smallest state. The state motto in Rhode Island is hope, and what I understand, and you know, you have to correct my pronunciation. Johannes en nomen ius. So is that correct? John is his name. Shoreline, we have about 40 miles of shoreline, but actually the coastline is 384 miles, which is really pretty similar to Puerto Rico. And we have about a third of the number of people within the state compared to Puerto Rico. And the highest point in Rhode Island is only 247 meters compared to your 1,300 meters. So you went out on high points in terms of how to get high. So this is Brown University, and this is what it's going to look like actually soon in the fall when all the leaves start to change color. This is actually a really pretty nice view, and this is the oldest. The building here is the oldest building on campus, and the Neuroscience is over a little bit over here. You can't quite see it, but here's the building that actually Carlos and David Scheinberg and Nadir and I are in. It's called the Sidney Frank Hall for Life Sciences, just to give you a sense of the place. What do we eat in Rhode Island? Well, Rhode Island's got this incredible food scene. I mean, it's just really wonderful. And it's a number of really pretty interesting places, but the state appetizer is fried calamari. And there's a number of really amazing people who grew up in Rhode Island. Viola Davis, who you will know as an Oscar-winning actress. She was also a Rhode Island School of Design grad. Other people, Jhampar Lahari, her books are just fantastic. If you haven't read her, she grew up in Rhode Island, and this is one of our presidents. Really dear president. Actually, she was president when David and Carlos and I were at Brown. So this is Ruth Simmons. Places where we visit, the coastline is just beautiful. And I'm just going to run through super quickly the things that we do. But there's an amazing East Bay bike path. And very importantly, with a Brown ID, you can travel around anywhere in the state free on public transport. Okay, so that's my hopefully just a 5-minute introduction to the state. We can answer any questions about that if you like. Then I'm just going to describe very quickly and then open it up for questions. But we actually have 2 graduate programs within the umbrella of the neuroscience graduate programs at Brown. One is we call the Neuroscience Graduate Program, and the other we call GPP, but that stands for the Graduate Partnerships Program. And that's in the first year, the 2 groups of students are together. And then the GPP students go to the NIH in Bethesda to do their graduate work there. We can talk a lot more about that if you're interested, but the cool thing is that we have 2 different graduate programs to choose from. They're both wonderful. And students do really, really well in both. There's a lot of training faculty at Brown. there's about 50 faculty who you can choose from within the neuroscience graduate program. There's a larger group of faculty who could potentially be trainers. And then at the NIH, the National Institute of Health in Bethesda, in their programs there, where they have the research labs, there's a very large group of neuroscientists, over 150 neuroscientists who could potentially be trainers. Right now, we have about 90 graduate students in our program, about 60 are at Brown, and about 30 are at the NIH in Bethesda. And our target class full recruitment is about 10 students for neuroscience and 5 for the graduate part. So about 15 students we're looking to recruit in the fall. We cover a broad range of topics from molecular to computation, and Carlos's talk I just think was wonderful and is really exemplary of the kind of research that people do at Brown, which is not just to do a single technique, but to really think very holistically about a problem, and then really apply a number of different ways to try and solve that problem across multiple levels. So I think Carlos, again, his presentation and his work is really just, I think, a wonderful example of the range of methodologies. We really care hugely about getting you into labs, doing research, educating, yes, I think it's a bit more of guidance really, but you're constantly learning. We teach you how to learn, we teach you how to critique, but there's also this very, very strong mentorship at Brown. We care about every single student who's in the program. And we have this wonderful, in the winter time, this neuropracticum opportunity to just be in the lab from morning to night and learn techniques, and several of us go to that. We spend 8 days there. We have breakfast together, we have lunch together, we watch movies together, and we're in the lab all the time. So here's a group of students who are at the neuropracticum. It's a really wonderfully cohesive group, and Nadira can tell you a little bit more about that. Okay, so finally, 2 more slides quickly, and then I'm going to turn it open to questions. We have the vast majority of our coursework is in the first year. It's intense, but you also have time to do lab rotations, so we get you into labs from the first day you're at Brown. And then, year 3 on, you're exclusively in your dissertation project, but that really starts in the beginning of year 2. We graduate people slightly below the national average, just over 5 years. And then money, the stipend. So the stipend is $36,000 right now. We give every student a moving allowance of $1,200. Students are very successful in the program, getting fellowships. And then there's all these opportunities to travel while you're a graduate student at Brown, go to annual meetings. Everybody goes to the Society for Neuroscience meeting when it's running, and we can talk more about that later. Obviously, it's virtual this year, but next year, everything will be different. Okay, and then just to end on this, we can have fun. And so actually the graduate students run these socials on Fridays, and they're still going on virtual, but you get a sense of there's always a theme. Our graduate students who graduate do really well, and we can talk a little bit more about that, but they span faculty, postdocs, industry, medicine, staff, scientists, government, consulting, science writing, education, and others. So again, we're really, really, we find it's very, very important to us that we consider each student as an individual, do a lot of individual mentorship, and really listen to you to see what it is you want to do and really help you get to where you want to be. So that is it. I'm done. Did I do okay, Nadira?
You actually finished right on time. That was exactly 10 minutes.
Great.
So I don't know if anybody has any questions. I know Carmen already introduced me, but I'm Nadira again. I am a 5th-year graduate student in the Brown NSGP program, which is one of the programs that Diane mentioned. It's the one where we're here the entire time. And I used to be a student in Carmen's lab. And well, I mean, I don't know. In a way, I'm still her student. I feel like we're forever. And she.
I'm proud of you.
We can share. We can share.
And she was actually, I know we were talking about the graduate program, but I know some people in Neuro-ID are not necessarily at the point where you would be applying right now. So one of the things that Carmen encouraged me to apply to before I was going to apply to graduate programs was the Leadership Alliance program, which is actually also centered at Brown. So for those of you who are really interested, and that's actually how I met Diane and David before I even got here. So for those of you who are interested in checking out the school, but you're not quite at the point of applying, that's also a great option to check out the labs.
It looks like we've got a bunch of questions, and
why don't you go on? I'm not going to read. You go ahead and decide which.
Oh, okay. And I did want to, David, do you want to say hi? Sorry, I meant to David Scheinberg.
I'll say hi. Hi.
David is our leader in the graduate program. He's making sure we say everything right. So there's some questions here. So Nadira, let's see. Oh, my student Roberta is in GPP and loves it. Great. Yay. How's the experience and how do you get in? Are there any prereqs? Do we have to do investigations as undergrad? So, well, you can start talking to us about, you know, what you've done already, but I'll answer quickly and then maybe Nadira can jump in or others. But you know, I think again, like so I should say that we don't look at the GREs. So we're really very interested in what it is, what do you want to do? Like, where's your passion? Are you, you know, what's your plan? Take a look at your, you know, what you've done as an undergrad. You know, what's your early entry into science? You know, why do you want to go to graduate school? Why do you want to go to neuroscience graduate program? So, you know, we would be interested in all those questions. And as I said, we dispensed with the GRE 2 years ago now, or at least a year ago. Yeah. So we didn't think that was a good predictor of success. It's well documented, it isn't. But, you know, other things are. And so we do like, obviously, to know that people have had some undergrad, some research experience, but, you know, so we can talk a little bit more about what your experience has been. And if you want to have more experience, we can tell you about how to do that. As Nadira said, there's some great programs. But again, we look at everybody as an individual and happy to talk to you about that. Anything to add, David or Nadira? Carlos?
I do. I was just going to say, in terms of research experience, was that, you know, aside from this Leadership Alliance, there's one thing that we also run is a prep program.
Yeah.
That, you know, if you feel that, say, for example, your grades weren't super strong and you didn't have much research experience, but you really want to go to grad school, then you could apply to our prep program. And actually, there's like 40 of them across the country, so I would recommend applying to many of them. And you come to Brown for a year, and you work in a lab, and you get a lot of mentorship, and you get help applying to graduate school, and as well as take a course for some topic that maybe you didn't have. And so that's another, if you feel that your application is not strong enough, or you apply and you don't get in, and they say, well, you're close, you know, a prep program is a good alternative, because you get a stipend, you get to work in a lab, and then you get help applying to graduate school. So I can send Carmen more information about prep programs in general.
And also in the as part of the program, you also get a graduate student mentor. I ended up mentoring somebody who participated in this program. So I think I definitely agree with Carlos that it's a really good opportunity. I will say that even, like, I know that some people who are interested in graduate school will do one of these programs just so they can also, like, maybe you do know you want to go to graduate school, but you want to try out working in something different. Oh, just for the record, this is like, a lot of times, I bet you've probably heard the word post-bac. This is effectively that, okay? Like, for those of y'all who are like there at that point. So, and also there are post-bacs at the NIH as well, and there are people who do their post-bac at the NIH and then apply to the GPP program. And that also helps them out a lot, especially if you're like, oh, you know, like, I've only done molecular work, and now I'm not sure, maybe I want to do something a little different. But so that's also a good chance to do that.
Yeah, but I would just say, just to wrap this up and then jump to another question, if you feel you're ready, go for it. So what we're saying is not, oh, well, you should think about it. We're saying, like, if you're ready, then definitely you should apply. But a good thing to do is to send your resume and send a note to the program director. It's currently David, or you can send it to me, because David and I run the training grant together. So we're both, all work in very close partnership, and then we can just have a chat. But I'm very much, I grew up in the British system, and I didn't do any coursework in graduate school, and I went straight from undergrad to grad. So, like, you can do that if you feel like you're ready. So lots of encouragement to apply. GPP. How does the GPP work? You do the work at NIH instead of Brown. Yeah. So the first year, everyone's together, right, Nadira?
Yeah, so for the first year, you like I'm sorry, I didn't mean to.
I want you to. I want you to answer.
It's actually really cool. So for that first year, you do all these activities together with your cohort, and we all have like a shared office. So it's great because you all get to go through everything together in that first year, including the fun activities that Diane mentioned, like the neuropracticum, which is also very unique and a very neat experience that I don't think many other places offer. And then after that first year, while you start doing your rotations, while you're done doing your rotations, then people will leave to the NIH for those who are in the GPP program, and then the people who stay at Brown then join their labs. The people who go through the GPP to NIH then do their NIH rotations, and so they later choose a lab. So you get to do rotations here in the school and then rotations over there. And I will say that I think some people actually get like a little sad sometimes when they're like, no, then you hang out with people and then they leave. But the school actually brings them back every other year to give presentations and for activities. And like in the case of like Roberto, so Roberto and I actually knew each other when we were both at the University of Puerto Rico. And then we were like hanging out here and we were like, you're here. And then now we actually still video chat with each other really often and talk to each other a lot. So even if people are like out there, you still have a lot of opportunities to hang out too.
Great.
Yeah, a lot.
Let's see. Yeah, did you want, are we just doing it straight from the chat, or are people going to ask?
We have, for example, it says, I noticed that the stipend from the GPP students is based on experience. Can you elaborate on what is that?
Yeah, it's based on research experience. So I actually don't, David probably remembers what it is, but so basically if you go into at the NIH, if you go in and you've had, like, let's say you've been a technician for like 3 or 4 years, then your starting stipend would be higher than someone who didn't have any research experience. So that's basically what it is. It's not a huge difference, I don't think. We've been increasing at Brown the stipend over the years, and it gets actually looked at very regularly to ensure that there's no to make sure that, one, we're competitive, so we get great students like everybody here listening, but also to be, to really be fair. Like, this is, you can be a student for 5 years and you need an income, and so we're really quite attuned to the importance of that. But that's the reason. It's a scale basically at the NIH.
And how about the MD PhD programs? Do you have that kind of offering?
Yeah, we have a small MD PhD program. The students are very good. Several of the students come into neuroscience. There are 3 or 4. David, do you want to?
Yes, we've had, in the last 5 years, we've had 3 MD PhDs do their degree with us. The class size in general is about maybe 4 or 5, and so a number of students have chosen neuroscience. They're great students, and for the most part, they do, as I mentioned in the chat, 2 years in they do their med school. Then they come to the grad program and are really just full-fledged members of our graduate cohort. They're often very motivated, as you might imagine, and sometimes speed through at a rate that's a little faster than our average on average. So, which is we're totally supportive of. And then they head back to do their last 2 years in med school. Right now, the Brown is working on their grad program is covered exactly as you've heard, and 2 of the 4 years are covered through the med program. So they're working on extending that and trying to make it so that it is competitive with the other MD PhD programs around the country. So, but it's a program that's really coming back, and I think Brown is taking it as really a jewel, because it's we get some really good students who are interested in both clinical and research.
Mm-hmm.
Okay, and then we have one of the people in the chat that she already has a PhD in linguistics, but she's interested in having a she says, if graduate certificates in neuroscience, do you give that, or kind of training similar to?
Over to David.
We do not have a certificate. We have had, we certainly have had doctors and surgeons come in and join our program and get another degree. We have actually had a student get a second degree before, but we don't have a special program that would be, say, accelerated or separate for someone who has a PhD. But we would, if someone were interested, certainly consider someone who even came in with those credentials, but they would be part of our cohort. So,
okay.
It actually opens up another unique feature at Brown that I hadn't mentioned. Many things I hadn't mentioned in the overview, but we do have at Brown the option to kind of do an open graduate program. So you can study in another discipline. So an example would be, if you were interested in teaching, you could get a teaching certificate through the teaching department, and that's not what you're asking, I know, because it's kind of the other way around. But if you were in the neuroscience graduate program, then there are other options that you can enroll and apply to, and enroll in a different kind of program, which gives you the opportunity and a little bit more funding to do extra time and get a certificate or a master's in a different area. And so a number of people like to combine things. Yeah.
And what is your deadline? Did I catch? No, what is the deadline?
It's generally the first week of December, but this year is very special, as you might imagine, as the graduate application portal isn't even open at this moment, as admissions across the university are being sort of considered exactly how we're going to do it. So we expect it to be sometime in early December, but it isn't actually set at this very moment.
Okay. Okay.
I saw that somebody, oh, sorry, Carmen.
¿Qué te iba a decir? Ajá.
So I saw that somebody asked about just generally, like, what would be a good thing, like what would be a good thing to do to apply for graduate school, like what would put you in a good spot. Mm-hmm. And especially if you still have a little bit of time, one of the things I would recommend, and this was, like, I'm telling you this, but this was straight up also the same thing that Carmen was like, this is what you have to do too. So I would recommend get yourself an amazing supportive mentor. Get somebody who can, like, vouch for you, who will work with you, and will give you a chance to do all these cool things in science. I feel like a lot of people get really wrapped up in stuff like grades, and the GRE doesn't even matter anymore, so you're good. But the really important thing is to do something you really like, to put in a lot of work into it and try to learn as much as you can. And just really try to figure out, okay, what is the thing I want to do for a few years, right? And I think after that, the other thing that helps a lot is doing these summer programs at other schools. So I mentioned Leadership Alliance. Leadership Alliance I mentioned it because it's specifically housed at Brown, like the main headquarters of it. But that program also allows you to apply to a bunch of other schools for summer research internships. So you actually would have the chance to go to some school that you really like, and I would definitely recommend you try out doing like a summer program for a few months and you get a little bit of that research experience, and that helps you also like meet other people who can also support you. And I think the last thing I would recommend is, besides that, like also if you are at a point, I know somebody mentioned something like, oh, hey, like what are my chances of getting into a graduate program if I don't have a lot of research experience? There are people that can get into programs without research experience. I've heard of the tales. I'm not sure necessarily how that really works because I have not been at the point of admitting people into a program since I am but a student. But I would, when you are in the graduate program, taking classes is really like the smallest thing of the program. A lot of it is really just you committing to doing a research project for any time up to like from like 4 to 6 years, right? So you want to be able to have, you want to succeed. You want to be able to like be ready. And that those postbacks and like the prep program, like Dr. Aizenman suggested, like that gives you a chance to see what research is all about and to be like, okay, can I commit to doing this for a long time, right? So I would, that would be like my 5 cents on that.
We do look at that we do weigh that quite heavily, you know, but again, you know, that every application is individual, but as Nadira said, we're all about research. And so to have some sense of, have you tried it? Do you like it? Can you weather the do you know what it is? You know, you won't get a Nature paper after doing 6 weeks in the lab. Like, you know, is that going to disappoint you? Like, so there's a sense of like, is it something you love? Like, do you have the passion and can you convey that? And we all do, and we all figured that out. And I think, again, as Nadira said, like I'm going to embarrass her, but like Nadira just like, every moment it's like she's just absorbing, you know, what people are telling her, and she's listening and contributing, and it's just like, it's just so much joy that like the passion is there. And so it's great. I mean, it's just great. So, you know, I think, I'm sure that many of you know, feel that because you're listening to this, but you know, that's kind of, yeah, it's kind of what we're, you know, is the passion there, and is the enthusiasm, you know, there. That's kind of important. And you don't know until you tried it, right? And then it just grabs you.
I meant to ask a question about student organizations for underrepresented groups like Latinos or African American. How is Brown in that realm?
Yes. Okay.
Oh, dear.
I'm ready for this. So, and Carlos is here too, so he also has a lot to contribute. So Brown University actually, like, they really have put in so many efforts forward for like diversity and like making sure everybody is like really like truly included and like they have their space. So I am currently part of SACNAS. I know a lot of you must be familiar with that. And there is a SACNAS chapter here at Brown, and we are very active, and we're doing a lot of different activities, anything ranging from like community outreach activities to activities within the school. We've set up so many different types of things. Like some of them have been asking Latina professors to give talks at the university, getting people to talk to a wide array of like professors if like they're like trying to find a mentor for themselves, like everything really. And it's like a great group. And the great thing is that like SACNAS here, I know that a lot of there's some variety between chapters, I think, at some schools, but here we know that we've tried to include everybody from all of STEM and even people from different departments. We're just like, just whatever, if you want to come over, come over. Brown has there is a grant here that is the IMSD grant, so the Initiative for Maximizing Student Development.
You got it.
I was on that grant, so I should know that. And they have such a variety of activities and workshops that they set up for the students. And it's open for everybody, but the main thing is that they really make sure that like students also like have their peer group that they can talk to and interact with. We also have NBRIT, which is an organization for students of color. And there's actually also a variety of groups for graduate students of color as well. There's literally a center for students of color. So like there's also like meetings that we have there, and we actually just like get together, and there's a huge community of just like everybody here at the university. I gotta say, I feel very included. So yeah. Oh, and actually, when Hurricane Maria was around, some of the Puerto Rican students actually got together to make like a separate organization so we could house students from the University of Puerto Rico, and they could actually like take classes here at Brown, and they wouldn't like miss their entire semester and stuff like that. So like we're also trying to be like really in tune to like what people need as well.
Wonderful.
Yeah.
And there's a question, though, do Puerto Rican students require the TOEFL for you guys?
Oh, not here. Sorry.
Well, we're asking because some schools do ask for the TOEFL. So
In general, if your coursework is offered, I don't know how it works at Puerto Rico, whether or not the classes are in Spanish. Yes,
I didn't know to take it though.
So
were not required, Nadira? Do you remember? No.
I did not have to take it to apply, although we had to take the GRE before. I'm hoping the TOEFL requirement has not changed, but at least when I applied, it was not a requirement. I don't think it has been.
I mean, we can confirm, but I don't remember ever.
I think from Puerto Rico
asking
it's
for a
not
TOEFL.
required, actually.
No, I don't. I've never done it, but we can triple check, but not to my knowledge.
Okay, because there's some too, and there's a cutoff.
I see.
No, University of Chicago requires the TOEFL.
It's definitely something that's not required by the graduate program, and so I don't know if the graduate school can override that. But I think that, yeah. So I'll ask. We should check, but I don't think so.
Okay.
I think more importantly, so our students in second year will be teaching assistants just for one course. It's not onerous. It's really part of the whole educational program. But there are some good, assistant. There's assistants, and so if you aren't comfortable, spoken, written language, there's ways at Brown to get that assistance as well. So I think that's perhaps the more important thing to mention. And there's also a fantastic teaching and learning center at Brown called the Sheridan Center for Teaching and Learning, and I've participated in that. Carlos has, I don't know if David has, and a very large number of our graduate students do as well. So.
Wonderful. Well, I think we're reaching our time to say goodbye. It has been a wonderful, wonderful experience to interact with all of you, staying until 6:30. I really appreciate it because I know time is valuable and you're very busy, but still you make time to talk to us, and we all appreciate this. And I'm going to hopefully see you again next week, because I am such a eternal optimist that we will continue to share science with you and others. And next time we'll have Harvard University and Dr. Data talking to students. So, yeah, we'll see everyone next week. And thank you once again, Carlos, Diane, David, of course, my dearest Nadira. And so have a great week, everyone. Thank you. Gracias.